welcome to everybody, and many thanks to the Data2 Foundation for having invited me to present our new data on ADA2, mainly focused on ADA2 and nucleic acid sensing, with focus on cellular mechanisms. Our presentation comes after the presentation from Ingrun that you have heard before, and that was speaking about molecular mechanism of implicating the crosstalk between ADA2 and nucleic acid sensing. So as you know, the ADA2, as you very well know, the ADA2 is considered an interferonopathy. Here you can see, for example, signs of increased interferon, type 1 interferon pathway, as well as other antiviral pathway in monocytes from the ADA2 patients in comparison to healthy control. And this is true from primary cells, so monocytes, but you basically obtain the same results when you knock out ADA2 in monocyte cell line. For example, here you can see in THP1, indicating that lack of ADA2 is inducing type 1 interferon production, a dysregulation of the type 1 interferon production. And but the mechanisms leading to this dysregulation are not known. And as you probably know, we have previously shown that ADA2 is a lysosomal protein. Here you can see an immunofluorescence staining of peripheral monocytes where nuclei are stained with DAPI, and LAMP1, that is a marker for lysosome, is stained with green, and ADA2 is clearly contained in the lysosomes. And as you have heard before me, we have recently shown that ADA2 is an enzyme that is able to convert adenosine to inosine into DNA molecule. Therefore, we propose this new term to define ADA2 as a lysosomal adenosine deaminase acting on DNA. And this is the first time that a lysosomal adenosine deaminase is with the natural targeting on DNA is described. But after this finding, then came the question if inosine in DNA can affect lysosomal nucleic acid sensing. And as you probably know, lysosomal DNA is mainly sensed by TLR9. Therefore, the next question we wanted to address was if inosine could be actually recognized by TLR9. So we have seen that ADA2 is able to convert adenosine to inosine in DNA molecule. But then we wanted to know if inosine is recognized at all by TLR9. And to answer this question, we designed some CpG analogs. That means that we converted some bases that are known to be essential for the recognition of the CpG gene to TLR9 with other bases. CpG is, for you to know, is the classic ligand of TLR9. And we converted G, so guanosine to inosine, cytosine, and adenosine, because the G, as you know, so the guanosine is the base that is normally recognized in TLR9. And our question was if what we call the CPI, so a CpG analog containing inosine, could or not activate TLR9, analogously to the CpG2216, that is the one with the guanosine. Therefore, to answer this question, we use cell line reporter. So these are HEX cells expressing human TLR9, and the activation of the human TLR9 is associated with the production of with an enzyme that can be then measured with a colorimetric assay. But basically what you can see here is that the CPI analog, so the CpG analog containing inosine, can be recognized as well as a CpG, indicating that inosine is efficiently recognized by TLR9 as efficiently as guanosine. And this we have seen in reporter cell line, but an analogous results could be obtained also from peripheral broad mononuclear cells to have, let's say, further evidence also from primary cells. And having answered this question, so being sure that inosine could be recognized by TLR9, then we wanted to see if ADA2 somehow could directly affect TLR9 signaling. And so to answer this question, we transfected the same cell line that we have used before with the ADA2 or with the empty vector. And then we again stimulated these cells with the different stimuli, but what you can basically see here is that in agreement with what we have shown so far, the overexpression of ADA2 is associated with the significant upregulation of the signaling downstream of TLR9. And this again was seen first in HEC cells, so in cell line, reporter cell line, but analogous results could be obtained also for primary cells. In this case, we stimulated the PBMCs with the different concentration of TLR9 agonist. And as you can see here, patients, so cells, PBMCs from the ADA2 patients resulted to be impaired in their production of interferon upon stimulation with the TLR9 agonists. We used PBMCs, but you have to take in mind that in this setting, in this experimental setting, stimulation of PBMC can be considered as an equivalent of stimulation of dendritic cells or plasmacytoid dendritic cells. And lastly, as you probably know, you know that ADA2 is mainly expressed with monocytes and macrophages. Therefore, we wanted to further address which is the impact of the absence of adenosine deaminase 2 on macrophages upon stimulation with the CpG or TLR9 agonists. And as you can see here, when you stimulate macrophages, human-derived macrophages in vitro with CpG, you obtain a very strong downregulation of CXCL10 that is a marker of nucleic acid sensing, and this can be seen both at protein level in the left panel, but also at RNA level, indicated that differently from what happens in dendritic cells, stimulation with the CpG in macrophages is actually an anti-inflammatory stimuli. And after having seen this impact on human healthy normal macrophages, we also analyzed what was the impact in the absence of adenosine deaminase 2. And as you can see here, basically lack of ADA2 is associated with a decreased downregulation of IP10 production upon CpG. So indicating that this immune regulatory pathway associated to stimulation with CpG in human macrophages is actually impaired in the presence of ADA2 deficiency. In conclusion, we can say that we have shown in our combined results that ADA2 act as a lysosomal adenosine deaminase acting on DNA, that adenosine deamination of DNA as well as ADA2 affect TLR9 signaling, and that ADA2 control CpG-mediated immune regulatory activity in human macrophages. And with this, I want to thank you and all the collaborators for your for the great help, and all for you for your attention. Thank you.